Okay, so thank you very much for having me here today. My talk's really going to focus much more on the biological side of things, looking at the regulation of the metabolic system in bacterial organisms. And I just put this picture up because I like it. It sort of shows it's a nice representation of the mess that's going on within the cell. Now, if this thing is going to work, it's not working. I'm out of battery, so I'll just click through by hand. Okay, so as you heard, I'm a group leader at the EBI, and what I want to do is really just acknowledge the contribution of different group members up front, particularly Ashwin Seshasai, who's a PhD student in the group and who's performed much of the work that I'm presenting here today. Also, a lot of this work is done in collaboration with Gillian Fraser in the pathology department and Madame Babu, who's a group leader at the LMB. So this is just a sort of a fun slide. So our work really focuses on genome scale analyses, and really there's a continued deluge of many different types of new data, and along with this deluge of data there have been a continuing expansion of the different kinds of omic terms or vocabulary used to describe these different data sets. So if you work down the list, you can see some familiar terms at the top. We've heard of the genome, the proteome, transcriptome, metabolome, and so on. That's all right. Interactome, we've heard of that. We keep going down, you start getting some weird ones like the functome and the unknome. That's the uncharacterized complement of the genome. Okay? And over here on the right, what we've got is a plot showing the rise in number of PubMed hits for some of these different terms. And you can see that the clear frontrunner has been the term genome. But also in the past few years, a few other terms have started to catch up, including proteome, transcriptome, and metabolome. And really the goal of our group is to take all these different types of data, depending on circumstances, and try to gain some understanding of regulatory systems. So our group is focused on the genome scale analysis of regulatory behavior of interesting biological systems. And much of this so far has focused on looking at transcriptional regulation. But we're also starting to look at other forms of control. And our interests are quite promiscuous within these boundaries, ranging from looking at the control of complex bacterial behavior all the way to trying to understand some aspects of mammalian regulation. What I'm going to do today is to focus on the bacterial work being done by Ashwin in the group. And as I said, this is sort of a collaboratory project with Gillian Fraser and Madame Babu. I'll let you guess which one Madan is on this slide. Okay, so a few facts about bacteria. They involve enormous numbers, which is very attractive to genomic scientists such as myself. So there are over 1 billion bacterial species. Compare that with a few thousand mammalian organisms. There are 10 to the 30 bacteria living on Earth. And this is the most disgusting one, 10 to the 14 bacteria living on each one of us, mainly in the gut. That's 10 times as many cells, bacterial cells, as there are human cells. So in terms of cell population, we're more bacteria than human. Of course, it depends on your level of hygiene as well. And the great sort of aspect of bacteria compared with eukaryotes, or more sort of advanced sort of humans and so on, is that rather than driving towards complex systems, bacteria really, their evolution drives towards efficient systems by simplifying their systems. The other sort of nice thing about them is that they're incredibly adaptable. On the one hand, you'll find bacteria occupying almost every type of habitat available on Earth. And even when you look at a single organism during its lifetime, it will adapt to a huge range of conditions such as acid or normal or heat or whatever, and it needs to adapt to all these different types of conditions. And these are two aspects that we explore when we look at their control systems. So what I'm going to do first is to talk about a completely computational project which we've been performing to look at how the metabolic system in bacteria is controlled. And then just the last sort of 10-15 minutes or so present a collaborative wet-dry project that we're performing that explores the multicellular behavior of bacteria. So we'll start off with small molecule metabolism. And just to make sure we're all on the same page, I'll go through some basics. So small molecule metabolism perhaps is the less sort of sexy system within organisms because it's been studied for ages. But it's important to reiterate how central it is to the survival of any organism. So any kind of core molecule that we find within the cell is processed at some point by the central metabolic system. And in small organisms such as bacteria, up to a quarter of its genome encodes for metabolic enzymes. So there's a huge emphasis on this system. And almost all organisms, ranging from bacteria all the way to humans, can serve quite a lot of the core metabolic processes. Now, of course, it's a complex system, but if you strip away everything and you just reduce it to its most important component, the central idea of the metabolic system is to ensure a stable concentration of ATP and AMP concentrations within the cell. Okay? So just maintaining a good energy balance within the cell. And this is done by the TCA cycle, shown here as a small cycle of reactions. But what we've got to remember is that this small set of reactions has to be coordinated with hundreds of thousands of others within the cell. Okay? And this is what we're interested in, because metabolism is an intricately controlled network that needs to balance the cost of protein synthesis, which is hugely expensive to small cells, and metabolite depletion, because you're using up important small molecules in the process, and the benefit of cell growth, cell division, and therefore population growth. And what we want to know is how this is controlled. Now, the textbook answer, and this is, I mean, you go into any textbook and it'll tell you that the metabolic system is controlled at two basic levels. First, you can control the level of enzymes, okay, by controlling the concentration of enzymes, mainly through transcriptional regulation. You can adjust the level of metabolites being processed. The other aspect is to control enzymatic activity. So once you have an enzyme, do you switch it on or off? And an example pathway here is the synthesis of the tryptophan biosynthetic, synthesis of tryptophan. You've got these different small molecules being converted to tryptophan by these intermediate enzymes shown here in purple. Now, the fun thing about this system is that tryptophan feeds back to control its own synthesis. So on the one hand, you've got tryptophan binding a transcriptional regulator, which goes to negatively regulate or repress the expression of these intermediate enzymes, and we'll call this indirect regulation, controlling enzyme concentration. And then on the other hand, tryptophan also binds directly to the enzymes involved in the first step of the reaction, and therefore inhibiting this step. And we'll call this direct regulation, and it controls enzymatic activity. So you see the two sides of concentration and activity being controlled. Now, there have been a lot of detailed analyses into how the metabolic system is controlled. In the 70s and 80s, metabolic control analysis became very trendy and popular. People did a lot of detailed analyses of famous pathways. Flux balance analysis is quite popular now, and here we're getting to genome scale analyses of how metabolic flux flows through systems to maximize output, and people have succeeded in integrating gene expression data and so on. But here the key word is the word balance, which means steady state. Therefore, you're not seeing any changes within the system, so we're not really interested in the control of this metabolic system. There was some very nice work done by Nam and Barkai a few years ago looking at the transcriptional regulation only in the yeast metabolic system. But here we missed out on understanding the regulation of the activity of the enzyme. So what we haven't had yet is a complete genome scale analysis of all different aspects of how you can control this system. And the two questions that we want to address are, why are there two methods of controlling the metabolic system, and how are these two methods of control deployed in the system? So what we did was to take a data set of the metabolic system from EcoPsych. Here what we do is represent the small molecules as circles, and the arrows represent the enzymatic reactions. We got about 600 metabolites in E. coli being converted to one another by nearly 800 reactions, and there are about 150 different pathways. Okay, and these pathways will be interlinked with each other. On top of this, we superposed a transcriptional regulatory network, and here we have about 500 regulatory interactions involving small molecule binding of a transcription factor and then onto the control of gene expression. And we also superposed direct regulation, or allosteric regulation, which involves direct binding between the small molecule and the enzyme. And here we're looking at 350 interactions. Now, the first important thing to notice is that this control is really occurring on a genomic scale. We're looking at a control of the entire system, not just some parts of it. And we're looking at nearly 1,000 different types of feedback interactions going on within the system. The number that surprised me most was that almost a third of all small molecules are somehow involved in controlling the synthesis of some other small molecules. So 1 in 3 small molecules are involved in this control system. And this is what you get when you sort of superpose everything, and it's a mess. And it doesn't mean much when we just look at that. Now, just before we start looking at the data, it's important to emphasize that the two types of regulation act on very different timescales. Direct regulation, or direct allosteric binding, occurs in the order of milliseconds. Indirect regulation, transcriptional regulation, occurs in the order of minutes. So there's a 10,000-fold difference in timescale between the two. And if we represent that graphically, that's a minute. You don't even see a millisecond. So it's a huge difference in timescale. So what we want to do is start off by looking at the regulation, these regulatory properties at a more detailed level. And one of the easy questions we can ask is, how many reactions does a single molecule control? And what we find is that when we look at the number of reactions per metabolite, what we find is that direct regulation, or allosteric regulation, involves pretty much one-to-one binding between the small molecule and the enzyme. So it's very specific, whereas indirect regulation, because of the activity of the transcription factor, you amplify the range of effect you have on the control of the target enzymes. But when we start looking at the pathways that they control, so we find that both direct and indirect focus their regulation on a single pathway rather than spreading themselves out across multiple ones. Now, given that direct regulation is specific within a pathway that may involve, say, 5 or 6 different enzymatic reactions, we want to ask, where does it occur? Now, here we have a schematic of a section of a metabolic pathway, and what we can clearly see is that these metabolic pathways are not linear at all. But what we can do is to divide these reactions into 2 major classes. One are the non-decision-making reactions. So a metabolite has no choice but to go forward. There's only one product it can become. Decision-making reactions, a metabolite can decide on what, there's a choice of several different products to become. So we ask, where does the regulation occur? And clearly for these allosteric regulators, the direct regulation, where you're focusing on a single reaction, the focus is pretty much on the direct decision-making reactions. So it's controlling the outcome of what products they become. Whereas transcriptional regulation acts on the entire pathway. Now, the common perception of feedback is that a particular product will feed back to control its own synthesis. So we asked, well, how often does this actually occur? Do metabolites actually feed back onto their own pathways? And in both direct and indirect regulation, actually the answer is quite surprising. Less than half of these small molecules are involved in controlling their own synthesis. The other half of these metabolites actually feed back onto other pathways, quite often functionally related ones. So what we're getting here is sort of a network of interconnected regulation between related metabolic functions. And we can see a few examples. For example, in allosteric regulation, what you might get are different types of nucleotides all focusing in on the same target enzyme. So you've got different products from different pathways all zooming in on a single enzyme. In the indirect transcriptional regulation, what you might get is a single metabolite binding to a transcriptional regulator that goes on to control many different related pathways. Okay? So you're starting to tie in the regulation of different pathways. Now, of course, we can look at these sort of local regulatory properties, but of course what we want to do is to go and see how these properties look on a global scale. And what we want to ask is how these regulatory methods are deployed in the entire system. Now, when we look at the metabolic system and we look at the topology of the network, we can see it's a bow tie structure. What do we mean by this? In the top half, you've got catabolic pathways that take in many different types of small molecules from the environment and break them down to assimilate them into the central energy production pathways. That's the TCA cycle, for example, that we saw in the middle of the big network diagram, that involves a very small set of core precursor molecules. And then in the bottom half of the network, you've got these anabolic pathways which take these core molecules and go on to synthesize the entire set of molecules necessary for the organism. Now, of course, you've got a few shortcuts. So, for example, if you've got a necessary product in the environment, instead of going through all the reactions, you can directly utilize it as a product in the pathways. And this is an actual representation of the metabolic system. You can see a clear bow tie structure emerging from the connectivity. So we ask, how is the regulatory system deployed in this network structure? And we can clearly see that there's a big difference in how they're utilized. We've colored the reactions according to the type of regulation going on. Direct regulation clearly focuses on anabolic pathways. Very few reactions are being regulated in this fashion in catabolic pathways, whereas transcriptional regulation, indirect regulation, occurs throughout the system. Now, if we go back to an earlier slide where we talked about decision-making reactions, we can see that all the decision-making reactions are going to occur in the biosynthetic pathways because you're taking a small set of molecules and synthesizing one of many different products. Okay? We can also ask where these metabolites feed back from. Now, the common perception is that feedback occurs from the end of pathways to the beginning of pathways. So we can divide metabolites into those occurring at the top of pathways, the middle, or bottom, and ask where they feed back from. In catabolic pathways, all the feedback is occurring from the top of pathways. Okay? And on top of that, most of this feedback, or feed forward perhaps, is a predominantly positive signal, so it's an upregulating signal. So you're taking the cell, well, we'll go on to that in a bit. When we look at the anabolic pathways in the bottom half, we see that almost all the feedback is occurring from the bottom of pathways. Okay? But for direct regulation, allosteric regulation, you're seeing some feedback from the middle of pathways as well. Now, when we, and the regulatory signal, the feedback is predominantly negative. Okay, so you're repressing. So in the top, you're feeding back from the top, and it's positive. From the bottom, it's negative. Now, much of this probably is selected by the specificity and concentration of the metabolites. Of course, when you look at the structure of the metabolic system, you're going to get the greatest specificity for regulation at either end of the metabolic system, because that's where the molecules are most diverse. Okay, so it makes more sense. Now, when we look at the concentration of small molecules, if you imagine that the central metabolic pathways involve 13 types of molecules, and from this selection of 13 molecules, we're synthesizing over 400 different types of molecules, you can see that you're going to require a much higher concentration of core molecules compared with all the products. And of course, there's going to be a difference in the concentration. And when you're doing some feedback from the bottom end of the pathways with direct regulation, where you've got one-to-one interaction between the metabolite and enzyme, you can't possibly regulate from the very bottom of pathways. You've got small concentration of metabolites. You need to take two steps, from the bottom to the middle, and then from the middle to the top. When you've got transcriptional regulators involved in indirect regulation, you can do this feedback in one step because you've got these intermediate transcription factors amplifying the effect. Now, we can also ask, where do these metabolites originate from? And they can, you can sort of partition them into two sort of sources of origin. A metabolite can either be imported into the cell from the environment, or it can be synthesized by some sort of reaction within the cell. And we can differentiate between these different metabolites. There are metabolites that are totally imported, totally internal, or hybrid, where you can get them from both sources. And when we look at the number of metabolites that feed back, we can see that almost all the feedback is being performed by metabolites that are imported into the cell, rather than those that are sort of totally internal. And what this means is that this regulation is being really driven by sensing the environment through the import of these different types of small molecules. What we also wanted to look at is how much overlap there is between the two forms of regulation. And we've colored those reactions that have both regulatory types, we've colored them in orange. Of course, you get very little overlap in the catabolic reactions, as we saw earlier, because you don't get indirect regulation. And here, what you get are transcription factors that sense molecules being transported into the cell and therefore upregulating the pathways that sort of metabolize them. In anabolic pathways, in the bottom half of the system, what you're getting are systems such as the one we saw at the beginning, such as tryptophan biosynthesis, where you're getting both indirect and direct regulation. Now, there's a small number of pathways that are regulated only allosterically, which comes as a surprise to many biologists because people imagine that almost every enzyme should be regulated at a transcriptional level. But there are some pathways that are expressed all the time and therefore do not have any transcriptional control. And an example of this is the one above. So there's a lot of sort of information there, so we can just put together what we've seen. So a cell imports a particular metabolite from the environment. Now, these cells won't be synthesizing all the enzymes that process these metabolites because it's a huge amount of energy being wasted on some small molecules that may or may not be there. But if you've got a prolonged signal of the presence of this metabolite, you'll get a positive upregulatory signal expressing those enzymes. You will then process those small molecules, feeding them into the small central energy metabolism pathways, which are then used to go and generate a whole array of different products. And here you've got decision-making forks where you decide whether to synthesize one product or another. Now, when you've got enough of something, perhaps because you found it in the environment and you can directly import it into the cell, this will raise the concentration of the product. You'll quickly shut down this pathway through direct allosteric means and divert the product to another pathway. And you need to do this quickly because if you're waiting around for the transcriptional regulators to come along, you've used up millions of core molecules. And if this signal is prolonged, you'll get slow downregulation of the entire enzymatic pathway, so you're saving energy on not synthesizing those enzymes. So what we're exploring here is a metabolic system that is intricately coordinated to balance the needs of cost and benefit of the cell. It is a genome scale network, so we do need to examine these things from a genomic point of view as well as a detailed point of view. And we can see that there are different types of regulatory interactions that selectively control different parts of the metabolic system. And this you'll miss out on if you're only focusing on transcriptional regulation, for example. And this is done by coordinating very different timescales, difference of 10,000, different specificities in terms of how these metabolites control their targets and enzymatic concentrations. And we feel that these findings probably hold true for many different organisms, including eukaryotes, because the transcriptional system holds true in yeast as well. And if we imagine how these cross-wiring feedback regulations sort of crisscross across different pathways, we can imagine how much evolutionary constraint it imposes on these pathways. Because if you evolve one part of the system, you directly impact on the use of another part of the system. And by looking at these, we can identify and manipulate perhaps some control points in the system. So I just want to spend the last 10-15 minutes describing a collaborative project we're doing to investigate multicellular behavior in bacteria. And this is I have to admit, I don't have many results to present here, but it's a nice project, and I just wanted to present it to you. So this work is really being done in collaboration with Gillian Fraser in the pathology department, and involves a couple of experimentalists in that group. Now, we can go back to looking at a few more facts about bacteria. And most people perceive bacteria as being single-cell organisms. Okay, you've got your liquid media and they're happily sort of feeding themselves. But in fact, most bacterial systems survive as colonies. And when I say colonies, we're talking about billions of cells together. And these colonies often produce some sort of multicellular-like behavior which optimizes growth and survival. You find different cells within a colony differentiating into different types of cells to perform specialized functions. By grouping together, it allows bacteria to colonize inhospitable environments that it otherwise wouldn't be able to. And one of the things that it does, for example, in biofilm formation, is that they construct a polysaccharide matrix that protects against desiccation and antagonists. And this is one of the ways in which bacteria can produce antibiotic resistance. And it is an incredibly widespread behavior among different species, and it's only really beginning to be appreciated. We find it occurring in the environment, so you can get biofouling of water, destruction of oil pipelines. You can get infections of medical implants and in artificial joints and catheters. And you can also find these colonizing behaviors in infections such as cystic fibrosis pneumonia. One of the manifestations of this behavior is called swarming, and it's the behavior that we're interested in. And what happens is that you get a single cell, we call it a vegetative cell. It will differentiate by elongating to over 20 times its sort of normal length, sometimes 50 times its length, overproducing flagella. They'll start moving together, so it allows, that's called the swarming behavior. And once they reach their destination, they'll divide and repeat this cycle. And you can see photos of these different cells. This is all the same species of bacteria. You've got the single cells, you've got the flagellating cells. They look completely different. This is a population of migrating cells, and they divide, and it repeats this cycle. Now, why is this, and this is sort of a photo of the two types of cells, just to show you the difference in size. Now, oh, and okay, here we've got some images. So what we have here is an agar plate. What they've done is to inoculate the center of this plate, and these bacteria have grown and swarmed by traveling in these sort of concentric circles. And I've got a video here that you can probably see. So they've got the front of bacteria swarming out. It travels much more quickly than it will be able to if it were a single cell. And it allows these organisms to reach inhospitable locations. It can't reach these locations without this. Now you see a wave of cell division following up, and it will eventually catch up with the front, and then they'll go and swarm again. So they're off they go. Okay? And you can look at a close-up of this behavior. This is the front. So these are the swarmer cells, so moving like mad, pushing the front forward. Okay? Now the reason that this behavior is important is that it causes infections. It allows bacteria to reach locations that they'll go and infect. One of the best studied models for this is called Proteus mirabilis. It's not a terribly sexy organism to look at, but it causes urinary tract infections. Okay, so in particular, it causes urinary tract infections in catheterized individuals. So if you're in the hospital, you're catheterized. After a month, you're almost 100% sure to have a Proteus infection. And this is a sort of this is a colony of bacteria on the end of a catheter. If you let it, it'll go up to your bladder, and if you let it, it'll go up to your kidneys. Okay? So what it does is swarming allows these bacteria to reach inaccessible locations, and it's a widespread behavior performed by many different bacteria, but with different efficiencies. Now, the key points about this behavior is that these bacteria upregulate the flagella genes that they use to move around in the media. So there's drastic upregulation of flagella. There's an inverse relationship between flagella and cell division. So when it's overproducing flagella and moving like mad, it can't divide. When it's dividing, it can't move around. And there's a great deal of cell-to-cell communication going on. Now, flagella control in E. coli and other bacteria is very well studied. You've got a transcription factor called LRP upregulating FLHDC. This is a sort of a heteromeric transcription factor which goes on to upregulate flagella synthesis. Okay, and this has been studied for years and years and years, and it's very well understood. Now, the inverse relationship between motility and cell division is very interesting because most cells, and in particular eukaryotic cells, cannot move and divide at the same time. And it's been cited as a possible driving force for the emergence of multicellular behavior, because organisms do want to be able to move and divide at the same time. So you've got flagella synthesis, you've got RCSB, which turns off flagella synthesis, but what it does is it allows one of the subunits in this FLHDC transcription regulator to go and upregulate cell division. So you can't have both occurring at once. Now, 20 years of molecular biology have uncovered 3 transcription factors that govern swarming, but such a complex behavior can't really be controlled by just 3 transcription factors. Okay, so it's very unlikely to be the entire story. And then the third point is cell-to-cell communication. And this is really an unknown area of this behavior. And there are many different sources of possible sources of communication, including physical contact between the cells, and in particular, the flagella sort of getting tangled up with each other perhaps senses the viscosity of the environment. And of course, a lot of small molecule signaling. But none of this is really understood. So the work we're doing really involves looking at sort of performing functional genomic experiments, including gene expression and chip-chip studies, to identify the regulatory interactions, superpose gene expression data in order to understand or uncover the active regulatory pathways that are controlling these different stages of the system. And one of the interesting aspects of this project is an interspecies comparison, because Proteus mirabilis, you see on the left, does this behavior wonderfully. You can almost not stop it, whereas E. coli very much struggles to swarm. And actually, you've got to use special agar imported from Japan for it to swarm. It's a very luxurious organism. So Proteus and E. coli have very similar genomes. They encode exactly the same transcription factors to very high sequence similarity, up to about 95%. But the outcomes of their behavior are completely different. So by comparing the gene expression and chip-chip data between these organisms, we hope to be able to uncover the sources of differences in these behaviors. So just to summarize, so this is a sort of a wet-dry project. We're sort of in the middle of the experiments, and we're not in a position to present any sort of results yet. But what we want to do is to identify novel mechanisms that balance motility and cell division and control intercellular communication. What we want to do also is that there are a lot of population models that dictate how, that sort of describe how these swarming movements occur. And what would be nice to be able to do is to link these molecular mechanisms with population level models. And of course, a lot of bacteria don't live in isolation, they live in multi-species communities. And their behaviors vary according to the mix of bacterial species you have. So one of the goals is to start looking at these multi-species colonies. We want to look at this because bacteria simply don't live alone. So just to finish off, acknowledge the main people who do the work. There's Ashwin, multicellular Ashwin, and then Annabelle and Jonathan. Ashwin did all the computational work, and he's in charge of all the computational work in the Proteus swarming project. Annabelle and Jonathan are doing all the experiments for the swarming work. And of course, this is also a collaborative work between Gillian Fraser, Madame Babu, and myself. And we've got other people in the group who also do other interesting projects, so perhaps I'll have an opportunity to present this on another occasion. Okay, so I'll take any questions if you have any. Thank you. Yeah.
It seems like in the direct to the anabolism,
yeah,
the regulation point should be due to the things that you can shortcut the metabolites around to. I mean, this assumes that the cell is only ever really in one state of dividing, which is perhaps not always really true. But you would have thought that the downstream products, the control of building stuff, can only really be influenced when you have the potential of a shortcut metabolite to come in. So have you sort of documented whether there's a higher amount of regulation on the ones where shortcut metabolites are present or not?
Yeah, so that's the whole point of the hybrid metabolites, those that are both synthesized internally and imported from and there is a much higher concentration of feedback from those types of small molecules.
3 or 7 of them or something like that. I can't remember.
Those are propensity. It's sort of a normalized value. It's not absolute numbers. Okay. Yeah.
I see.
So, but that's a very good point. But there are interesting ones where they're totally synthesized internally.
Right.
And those are the ones that regulate the pathways that are not transcriptionally controlled. So you overproduce something and it sort of stops. And then
The other sort of question I had in my head is, have you or anybody else thought about so this flux balance kind of framework works if you assume steady state, and it's kind of a vector of fluxes, right? There's a set of vector of fluxes of which one will be optimal for the
cell growth, yeah,
for whatever criteria you put down to it. Now, presumably, you could think about each state being a set of, for every input state, you have a new set of fluxes.
Yeah.
Is that correct? Have you thought about kind of families of flux balance models in this framework or something like that? Is that the right way to go? Or is that going to be not right?
I have to admit, we haven't come to grips with flux balance analysis. One of the reasons is that it's very difficult to come by good data measuring sort of actual fluxes. So a lot of it's sort of theoretical calculations, and then people have problems reconciling them, say, with gene expression data and so on. So it's not quite clear how to go about that. I mean, obviously that would be a very nice thing to do.
Okay.
But it's something that we haven't sort of managed at the moment. Yeah. No? So we have an early lunch then. Yeah.
All right, let's thank our speaker again.